Good afternoon, everyone. So the next topic that we are going to study under Autacoids, it is prostaglandins and thromboxane. Okay. So these are the contents of my presentation. So first of all, we are going to see the prostaglandins, the historical perspective of it. And then we are going to study the chemistry and biosynthesis of the prostaglandin as well as thromboxane. Then we are going to study the pharmacological actions of prostaglandin and thromboxane, and lastly we will be studying about therapeutic uses of the prostaglandins. Okay, so about this prostaglandin and thromboxane, you have already heard in the chapter the inflammation and tissue repair, which you have studied in the course, that is pathophysiology. But in this particular course, that is Pharmacology-II, we will be studying more about the pharmacology of these particular substances and their various effects these substances produce on the various organ systems, and also we will be studying in which clinical conditions the analogs or the antagonists of these substances they are being used. Okay, so we won't be going into the details that why these substances are being used in these conditions, but as we will be studying the pharmacological actions, you will get to know that the utility of these substances in the clinical practice. Okay, so let's start with the first thing, that is the prostaglandins and the historical perspective of it. So in 1930, two scientists, that is Kurzrock and Lib, they demonstrated the activity of human semen on isolated strips of human uterine muscle, and this was confirmed by another scientist, that is von Euler, in 1935, who demonstrated a substance present in the extracts of the human seminal fluid which cause the contraction of the isolated intestinal and uterine muscle, and also it caused the vasodilatation. So this substance, it was named as prostaglandin because of its probable origin from the prostate gland. But later on it was found that it is not only limited, or it is not only present in the prostate gland, but it is present in some other organs and some other tissues also. Okay, so another scientist, Bergstrom, who got Nobel Prize in 1982, and his associates, they showed that the various prostaglandins, they are closely related derivatives of a lipid soluble prostanoic acid. And on the right there is a structure of the prostanoic acid. Okay, so chemistry and the biosynthesis. So first of all, before we actually study about the prostaglandins, about the thromboxane, about the leukotrienes, we need to first of all understand some basic things about the fatty acids. And the fatty acids, those who are beneficial to our body, and the fatty acids, those who are actually harmful to our body. So in detail we are going to study. As such, this all theory, it won't have any significance in exam point of view, but for the understanding purpose this theory is must. So we will just study this theory so that you will have better understanding of the things that are yet to come. Okay, so fatty acids which are occurring in natural fats, they are mostly straight chain aliphatic compounds with even number of carbon atoms which are connected by CC bonds, carboxy group at one end and methyl group at the other. So the fatty acid, it may be saturated. So when we can call it as saturated fatty acid, the short form is SFA. Please remember the short forms. When there is no double bond present in the structure, we can call such structure or such fatty acids as saturated fatty acids. So opposite is true for the unsaturated fatty acids. The abbreviation is UFA and it shows presence of double bonds in it. Okay, so UFAs may be monounsaturated fatty acids, that is MUFA, with a single double bond. Okay, or it can be polyunsaturated fatty acids. So when we can say it is polyunsaturated for fatty acid, when it contains two or more double bonds in it. Okay, so the names of the SFAs end in ANOIC. Okay, so the suffix is anoic. So the prostanoic acid, the structure which we have seen, it is a saturated fatty acid. And in case of UFA, the suffix is ENOIC, that is enoic. And the example is icosa pentanoic pentanoic acid. Okay, so this is matter of concern, that is icosa pentanoic acid. Okay, so next, fatty acid, they serve many functions in the body. So the first function is they are responsible for energy production. Then they play part in electrical insulation. Then they are responsible for the transport of the plasma lipids. Also, they can help in maintenance of the cell membrane integrity. If you remember the bilipid structure of the plasma membrane, it is the fatty acids, they are one of the components of it. Okay, so they also help in proper development of the CNS. Then they can help in participation in cell communication and also help in oxygen transport, and also it can help in regulation of inflammation. Okay, so animals, they can synthesize the SFA, that is saturated fatty acids, from the carbohydrates and some amino acids. However, unlike plants, they cannot synthesize two PUFAs, that is polyunsaturated fatty acids. And which two PUFAs the animals cannot synthesize? The first one is linoleic acid, the short form is LA. Again, remember the short form. And the second PUFA, that is alpha linolenic acid, that is alpha LNA, which are therefore essential in animals. Okay, so using these two, the animals synthesize all other UFAs they need. Okay, so the all other unsaturated fatty acids, they can be prepared from these two basic, that's why they are called as essential. Okay, and the example of other UFA, that is given as the arachidonic acid, and this is the very much important. Okay, now you will understand why I'm saying that arachidonic acid is important once we see the all of these substances, all of these autacoids, they get generated from this particular UFA, that is arachidonic acid. And this arachidonic acid, it is important constituent of the cell membrane. Okay, so next, icosanoids, they are oxygenation products which are derived from the 20, that is icosa, okay, carbon UFAs. So all of these, the prostaglandins, the leukotrienes, thromboxanes, they are all icosanoids. And why they are called as icosanoids? Because they consist of 20 carbon atoms in their structure. That's why they are called as icosanoids. And what are they? Actually, they are unsaturated fatty acids. All of them, they are the unsaturated fatty acids. So we have dihomogamma-linolenic acid, that is DGLA, then arachidonic acid, and the icosapentanoic acid. So all of these, they come under the category of icosanoids. Okay, so this DGLA, arachidonic acid, and the EPA, all of them, they are collectively called as icosanoids. And what they are, they contain 20 carbon atoms and they are unsaturated fatty acids. Okay, so they are ubiquitous both in animals and together with precursor oil in various plants. Ubiquitous means commonly found or found in large quantities. Okay, so arachidonic acid, it is the major source of prostaglandins, that is icosanoids in the mammal. So our matter of concern or our substance of concern over here, it is the prostaglandins, and these prostaglandins we get from the arachidonic acid, which is nothing but an icosanoid. Okay, so the three respective series of the icosanoids we have. The first series is PG1, second is PG2, third is PG3. Okay, and these three series, they are defined by number of double bonds in their side chains. Okay, so PG1 and PG2 series, they are synthesized from La. Okay, so which is an omega-6 PUFA, and its derivative GLA via DGLA. I have asked you to remember the abbreviations, so these abbreviations can be utilized over here, and that's why it is a G1 series. And from AA, that is arachidonic acid, we get G2 series. Okay, so PG1 series we get from this, from the La, and the PG2 we get from the AA, that is arachidonic acid. PG3 series, it is derived from the alpha LNA via EPA, which is an omega-3 fatty acid. Okay, and docosahexaenoic acid, that is DHA. It is shown in this particular diagram itself that this alpha LNA, then in presence of this omega-3, that is EPA, and also presence of DHA, we get PG3 series. Then this PG1 and PG2 series we get from the LA, omega-6, through the GLA, through the DGLA we get PG1, and through the AA, that is arachidonic acid, we get PG2. Okay, so what is the importance of this series, that we will see in next slide. So almost all the tissues, they are capable of synthesizing these lipid derived autacoids. Their biosynthesis is triggered by trauma, infection and inflammation. Please remember in which condition these particular substances, they are getting synthesized. In case of trauma, that is tissue injury, in case of infection by some bacteria, then in case of inflammation, these particular substances, autacoids, they will get synthesized. Okay, so during their synthesis, the arachidonic acid is first released from the cell membrane phospholipids by action of an enzyme, that is lipases. Okay, so it is then oxygenated by 4 separate routes, which include first route, that is cyclooxygenase route, that is COX route, and second is lipoxygenase route. that is LOX route. Okay, so this COX route, it gives us the prostaglandins, and the lipoxygenase route, it gives us the leukotrienes. Okay, and from where we get it? From the arachidonic acid. Okay, so this particular chart, it shows how we get the prostaglandins from the arachidonic acid. Okay, so dietary essential fatty acids, they are incorporated into the membrane as membrane phospholipids. This membrane phospholipid, in presence of phospholipase A2, it gets converted to the arachidonic acid. And arachidonic acid, by four ways, it can give variety of product. The first one that we are going to study today, that I will just elaborate, that is in presence of cyclooxygenase. That is, there are two isoforms of this particular enzyme, that is COX-1 and COX-2 enzyme. It gives rise to an intermediate, that is PGG2. Okay, so this PGG2, again in presence of the same enzyme, that is COX-1 and COX-2, can give rise to the PGH2. Okay, and this PGH2, in presence of PGI2 synthase, we get PGI2 or the prostacyclin. Okay, so PGH2 can also give rise to the PGF2 alpha, then PGD2, then PGE2, and in presence of TXA synthase, it can give rise to the TXA2, that is thromboxane. Okay, so in which condition it will give rise to this PGI2, it will give rise to PGF2 alpha? It depends on the location where this particular substance is present. Okay, depending on the location, the formation of the prostaglandin, it depends. Okay, so we are going to elaborate it further into when we will study about the pharmacological actions of it. Okay, then arachidonic acid, which is a PG2 series fatty acid, it is a constituent of the phospholipids which are normally present in the human cell membrane. We have already seen that arachidonic acid we get from the membrane phospholipid only. Okay, so it gives rise to derivatives, that is PGs of the PG2 series and the LT, with marked pro-inflammatory activity. Okay, so whatever prostaglandins we get, they have inflammatory effect, that is, they promote the inflammation. Okay, so they have been incriminated in pathogenesis of chronic inflammatory disorders such as psoriasis and the rheumatoid arthritis. Okay, so in these particular diseases, it is found that these particular arachidonic acid metabolites or the products of it, they play very important role in the pathogenesis of it. Okay, so icosa pentanoic acid and the docosa hexanoic acid, both omega-3 polyunsaturated fatty acid, they are present in fatty sea fish. When they are ingested regularly, they can partially replace the arachidonic acid of the PG2 series with the less pro-inflammatory eicosanoids of the PG3 series, that is TXA3 in the tissue. Okay, so it may benefit the patients with chronic inflammatory diseases such as RA and the psoriasis. RA stands for the rheumatoid arthritis, and also it may help to protect from the atherosclerosis. So what all this means is that arachidonic acid by itself, when it undergoes certain pathways, that is COX pathway and LOX pathway, gives rise to the pro-inflammatory mediators. But if we consume the food which is rich in this particular omega-3 PUFAs, then these omega-3 PUFA, they will substitute the arachidonic acid from its location, that is from the membrane phospholipids, and as it partially replaces arachidonic acid from its location, what will happen? The chances chances of the atherosclerosis, they will decrease, and also it will benefit the patients who are suffering from the chronic inflammatory conditions like the rheumatoid arthritis and the psoriasis. Okay, so that's why it is many of the times you may have seen in the advertisements of some of the popularly used edible oils, they promote these omega-3 polyunsaturated fatty acids. They say that their oils, they have this. So you should be able to correlate the meaning of that advertisement with the benefits that they give over here. Okay, you should be able to tell like authoritatively that why we need to go for such oils, why we need to avoid certain oils like the coconut oil. Okay, so other dietary sources of the omega-3 fatty acid, they are the flax seeds, the nuts, particularly walnuts and hazelnuts, then dark green leafy vegetables and the soybeans. Okay, so consumption of all of these food items, it is actually beneficial in conditions like the rheumatoid arthritis and the psoriasis, as well as those who have predisposition to the atherosclerosis like the obese patient. You should suggest that they should have this particular food items in their normal food. Okay, so that they will get benefit out of it. Okay, so under stimulation by the mechanical, thermal, chemical and bacterial insults, the PGs, they are released in large amounts into the body. So both PGE2 and PGI2, they are potent vasodilators, and also they cause hyperalgesia. Okay, so PGE2 is also potent pyrogenic substance. Pyrogenic means what? It causes pyrexia, that is it increases the body temperature, it causes the fever. Okay, so in association with other mediators, the PGs, that is prostaglandins, they play an important role in the development of the inflammatory response. Okay, so pharmacological actions. So the prostaglandins, they produce their effect by acting on the PG receptors, and it produces its physiological effects by acting on following systems. So these are the system. First one is smooth muscle, second is gastrointestinal system, third is cardiovascular, fourth one is kidney, fifth one is reproductive, sixth one is CNS, seventh one is the platelet aggregation. It also have role in this. Then it has a role in inflammation, but obvious, and also some miscellaneous roles it plays. Okay, so these are the various receptors you can see onto which the prostaglandins, they act, and also the physiological ligands, they have been enlisted, and this indicates that the PGI2, it has more efficacy or more affinity towards this IP receptor when compared to the PGD2. Okay, and PGI2 produces more profound effect onto the this IP receptor when we compare it with that of the PGD2. Okay, and distribution, where it is located, and the general physiological effects, all of these receptors they produce. Okay, and also the signaling system. So signaling system, you can see that all of these receptors, they are the G protein coupled receptors. Okay, so you can refer this particular diagram for your own sake, otherwise it is not necessary to remember all of these receptor types. Okay, you can just go through it. Okay, so smooth muscles. So On smooth muscle, PGE2, PGF2 alpha and the TXA2, they stimulate human myometrium and also increase the intestinal motility. So PGE inhibits the tone of tracheal and bronchial muscles and thus have the bronchodilation action. Okay, so this next is the gastrointestinal system. So prostaglandins, they are distributed throughout the gut through their concentration varies in different parts. Okay, so the PGE2 and PGI2, that is prostacyclin, which is synthesized by the COX-1, they inhibit the gastric acid secretion. Okay, and they enhance the mucosal blood flow and thus have a cytoprotective effect on gastric and duodenal mucosa. Both PGE and PGF, they cause contraction of the longitudinal muscles of the gut. Okay, so further, the prostaglandins, they stimulate intestinal fluid secretion and they cause diarrhea when given orally or parenterally. This suggests their possible role in certain types of diarrheas. Okay, so what is important over here, the first action is that it's the inhibition of the gastric acid by the prostaglandins. Okay, so when we have studied about the pathophysiology of the peptic ulcer disease, that time I have told you about this particular role. There are some protective factors and there are some aggressive factors. So the protective factors over there, it is the prostaglandins. Okay, and in case of peptic ulcer disease, the amount of prostaglandin, it decreases in that stomach region, and that's why there is excess of gastric acid secretion, there is decrease in the protective factor and thus the ulceration. Okay, so on cardiovascular system, PGE2 and PGI2, they are potent peripheral vasodilators. They mainly dilate the arterioles and post-capillary venules. The blood flow to the most organs increases because of this particular action. Okay, so the TXA2, that is thromboxane A2, it is a potent vasoconstrictor substance. The PGF2 alpha, however, is neutral in this respect in humans. The PGE1, that is alprostadil, is a potent relaxant of the smooth muscle of the ductus arteriosus and thus it preserves the ductal patency in the newborn. Okay, so a specific blood vessel which is responsible for the circulation in the newborn, it needs to be remain patent still till complete development happens in the newborn. So patency means what? It keeps it open, okay, so that the blood can flow through it, through this particular ductus arteriosus. Okay, and there this PGE1 alprostadil, it is a name of drug, it keeps that open. In some newborns there is a problem of the closing of this ductus arteriosus, and that's why we need to give the drug so that it will remain open and it won't create the further complications. Okay, so kidney. So the kidney cells that synthesize the prostaglandin contains mostly the COX-1 enzyme. So although the prostaglandins, they are not involved in maintaining normal renal blood flow, their local production is important in maintaining the blood flow in compromised kidney. Okay, compromised kidney means some abnormality is there with the kidneys. Kidneys as such, we know they are highly perfused organ. Highly perfused means there is a lot of blood is flowing through the kidneys or to the kidneys, and whenever there is obstruction to the blood flow, these particular prostaglandins, they play very important role in maintaining the blood flow to the kidney. Okay, so the PGE2 and the PGI2, they cause diuresis. Also they cause natriuresis and also cause kaliuresis. Kaliuresis, it is excretion of the potassium. Okay, and they have this direct action on the renal tubules, and that's how they cause the diuresis, natriuresis and kaliuresis. The PGE, it inhibits water reabsorption, which is induced by the antidiuretic hormone, and it also inhibits chloride reabsorption. So you should understand this PGE, it acts at the collecting duct. Why? Because the site of action of the ADH is the collecting duct, and as PGE inhibits the water reabsorption by the ADH, the site of action for the PGE is the collecting duct. Okay, so on the reproductive system, apart from their stimulant action on the uterine smooth muscle, the prostaglandin exert other actions on the reproductive system also. So in animals, the prostaglandin, they cause regression of the corpus luteum, which is nothing but the luteolysis. Okay, and also they cause reduction in secretion of the progesterone. This can prevent the implantation of the ovum. Okay, so they actually prevent the pregnancy by this particular action, because implantation of ovum is essential for the pregnancy, and as they prevent it, they prevent the pregnancy. Okay, so both COX-1 and COX-2, they are expressed in uterine epithelium at different times in early pregnancy. Okay, so COX-2 may play a role in the birth process, and its excessive activity may be responsible for the premature labor. So labor pains which occur prematurely, they are because of the excessive activity of the these particular prostaglandins. So the function of the prostaglandin which is present in the semen, it is not clear, although it was first found that the prostaglandins are present in the semen, but we don't know exactly what role they have in the semen. Okay, so onto the CNS, the role of PGs as neurotransmitter, it has been suggested. Okay, so the COX-1, it is distributed in neurons throughout the brain, being most prevalent in the forebrain region. COX-2, it is also expressed constitutively in a few areas. Constitutively means it is present by itself. Okay, so it is not situational presence of it, it is present by default. Okay, so the PGs, they are probably involved in central mechanisms that cause hyperalgesia. Hyperalgesia is like hypersensitivity to any stimulus. Okay, so one example I can quote of that: if you just touch any pointed thing to your skin, you won't be very sensitive to that particular that pointy thing. But in case of hyperalgesic condition, that small touch by that pointy thing also will elicit a very exaggerated response. So that is hyperalgesia. Okay, so further, PGE2, it is implicated in causation of fever, very common, that is PGE2 mediated action. Then in platelet aggregation, the only isoform detectable is the COX-1. So the prostaglandin endoperoxide and the thromboxane A2, they cause platelet aggregation and vasoconstriction, while PGI2, that is prostacyclin, which is found in the vascular endothelium, is a potent inhibitor of the platelet aggregation and it causes vasodilation. Now understand, there are two types of substances. one type of substance, it is causing the platelet aggregation and the vasoconstriction, and the other type of substance, it is producing the exactly opposite effect, that is it is preventing the platelet aggregation as well as it is causing the vasodilatation. Okay, so the Pg endoperoxide and the thromboxane A2, it promotes the aggregation, and the PGI2, that is prostacyclin, it prevents or inhibits the aggregation. So when platelets are damaged, they release Pg endoperoxides which cause aggregation of the platelets. But however, vascular endothelial PGI2, it has anti-aggregatory effect. So when the endothelium also gets damaged, what happens? The PGI2 synthesis, it decreases, and as PGI2 synthesis decreases, the amount of PGI2 present in that particular area, it also decreases, and as amount of PGI2 decreases, the activity of the endoperoxide and the thromboxane A2, it becomes dominant, and that's why the platelets aggregate whenever there is endothelial injury. Okay, so in inflammation, the presence of PGE and the kinins in inflammatory exudate, it suggests their role in the genesis of inflammation. Okay, so we will be studying about kinins in detail in next few chapters. That time we will discuss about this. So COX-2 is clearly associated with inflammation. Okay, so this it is basically whenever there is inflammation, the prostaglandins which get generated over there, they are because of presence of or the action of the COX-2. Otherwise the COX-1 enzyme, it has dominant action or it has dominant effect in production of the prostaglandins. So selective COX-2 inhibitors, they may control inflammation without affecting beneficial effects of the PGE on the stomach. So keeping this in mind that the COX-2 has a dominant effect in the inflammation, specific COX-2 inhibitors, they were synthesized, thinking that it will be helpful in conditions and it won't have the adverse effect the non-specific COX inhibitors have. But unfortunately, these COX-2 inhibitors, they had some other adverse effects, and these adverse effects were major. That's why these selective COX-2 inhibitors, they were withdrawn from the market. Okay, so miscellaneous actions. PGE1 and the PGE2, they block lipolytic effect of the adrenaline, ACTH and the glucagon. They also produce pain when it is applied to the exposed blister base. Blister, whenever you have like a burn wound, that time a balloon like situation or balloon like condition happens onto the skin, that are nothing but the blisters, and when we apply these prostaglandins at the base of these blisters, it can cause the pain. Okay, so some prostaglandins, they are potent bone resorbing agents which act by stimulating osteoclast. So we have talked about this particular one. Osteoclast are the cells that dig up the bone. That is, they remove the calcium and phosphate which is embedded into the bone matrix, and they make it porous, and that's why this excess activity of the osteoclast, it is responsible for the osteoporosis. Okay, so experimentally, the bone destruction induced by the implanted metastasizing tumors in rats and rabbits, it can be reduced by the prostaglandin inhibition by aspirin or the indomethacin. So this bone destruction we can prohibit or we can inhibit by giving the prostaglandin inhibitors, the drugs like aspirin and the indomethacin. Okay, so the PGE2, it also stimulates adrenal steroid production, and also it causes release of insulin. Okay, so therapeutic uses of the prostaglandins. It can be used as an abortifacient, that is cause the abortion purpose. The prostaglandins can be used, and also they can be used for the cervical ripeness. So they can be used as a gastric cytoprotectives in treatment of the peptic ulcer. They can be used, and the specifically drug that can be used is misoprostol, that is PGE1. So they can also be used in erectile dysfunction. So the drug which we use, that is alprostadil, and it is injected intracavernously. Okay, so in the penile veins this particular drug is injected. When it is injected, it increases the local blood flow, and thus it causes erection. Okay, so for maintenance of patency of ductus arteriosus, we have already seen the PGE1 analog, that is alprostadil. It can be given intravenously. So in treatment of primary pulmonary hypertension, the PGI2, that is epoprostenol by IV infusion, is effective. Please understand one thing: the prostaglandins in their natural form we cannot use because they are highly unstable preparations. Instead, what we do, we use the analogs of it. Analogs means a look-like substance. Okay, so when I say PGI2 into bracket epoprostenol, this epoprostenol, it is nothing but an analog of the PGI2, that is prostacyclin. Okay, and it is to be given by the intravenous infusion. Okay, next is it can be used in treatment of the glaucoma. The drug which we can use, that is latanoprost. Then also we can use for treatment of the peripheral vascular disease, the drug which we can use, that is epoprostenol. And also post AMI, that is acute myocardial infarction, to reduce the infarct size, we can use iloprost. Okay, so these are all the therapeutic uses of the prostaglandins. Thromboxane, as such, it doesn't have any therapeutic use, that's why it is not mentioned over here. Okay, so with this we have finished with the chapter, that is prostaglandins and the thromboxane, and the next chapter that we are going to start in next lecture, that is leukotrienes. Okay, we will mostly finish with the leukotrienes in like 15-20 minutes because it is very small topic, and then we will start with the next, that is mostly the angiotensin or the bradykinin. Okay, so that was all from my side. Thank you. Okay.